Good afternoon, everyone. My name is Brenda Padilla. I'm a second-year master's student here at Northern Arizona University, where I work with my PI, Dr. Robert Keller. And my work focuses on electrospun scaffolds with rhanolipids to treat depleted uranium-contaminated wounds. Now, my work comes from the use of uranium, and uranium is a naturally occurring heavy metal commonly found in the Earth's crust. And historically, uranium has been used for fuel, for nuclear power plants, and in the military use for anti-armor ammunition. And uranium can be found all over the globe, but right here in northern Arizona. And so located in the American Southwest, we have the Navajo Nation, where located in this area, it was swept into this mining boom, and as a result, there are now over 500 abandoned uranium mines scattered along this land. And here I have a map illustrating the Navajo Nation in yellow, and the red dots indicate the abandoned uranium mines. Now this population, 30% of it resides in rural communities, and they rely on unregulated water sources. And what this means is that these water sources are hardly, if ever, checked by government officials. And a study found that 12% of these wells exceeded the maximum contaminant level of uranium, meaning that whether this population knows it or not, they are ingesting this water that exceeds the safe drinking standard. And so internal uranium toxicity can lead to serious health issues, including but not limited to kidney disease, lung, and bone cancer. And previous studies from our lab found that exposure to uranium has negative effects on the wound healing process. And however, there lacks a healing therapeutic to counteract this issue. And so in this study, we are using rhamnolipids, which are bio-inspired surfactants produced by synthetic eco-friendly methods. And uraniums have demonstrated this ability to bind to metals of environmental concern, like uranium. And here we have the chemical structure of the mono-rhamnolipids that we use in this study. And we want to see if we can implement this metal binding ability from these rhamnolipids into the wound bed. And so we primarily started off with a cellular migration assay, which is an in vitro wound model that is created after a mock wound. And here I have the general process of that cell migration assay. And then to the right of that, I have images of what these skin cells can look like underneath the microscope after undergoing this mock wound, where we watch how they grow back together. And so here we have the results of our cell migration assay, and I'll bring your attention to the left. Here we have on the y-axis the percent closure rates, and on the x-axis we have our different treatment groups, that being our control, our low rhamno concentration, and our high rhamno concentration. And here I have highlighted in red that there was a significant increase at that 12-hour mark with the cells in our high concentration of our rhamno in comparison to the control. And then to the right, we have a similar orientation with that percent closure rate being on that y-axis and our different treatment groups on the x. However, I do want to point out that we expose these cells to environmentally relevant levels of uranium. And again, we saw that similar trend highlighted in the red, that at that 12-hour mark, our cells were completely closed in comparison to the control, regardless of uranium being present. And so we wanted to test the biocompatibility of our treatment, and we did this by using a Presto Blue and Cyquant Direct assay, where Presto Blue measures metabolic activity of the cells, and Cyquant Direct measures living cell DNA content. And here we have the results of our Presto Blue assay, which again measures metabolic activity of our cells. And on the y-axis, we have the relative fluorescent units measured, and then on the x-axis, we have our different treatment groups. And here in the big red box at the top, we have seen that, once again, there was an increase in metabolic activity with our cells with our low and high rhamno concentration in comparison to our control. However, I do want to point out that we implemented a max concentration of our treatment, that being 100 micromolar, and we saw that there was a significant decrease in metabolic activity. And this could tell us a lot about the future usable concentrations of rhamnolipids in future studies. And here we have the results of our Cyquant Direct assay. Again, similar orientation with the relative fluorescent units in our treatment groups on the x-axis. And the Cyquant Direct measures living DNA content. And again, we saw with, like in the Presto Blue, that there was another significant decrease in living DNA content with that max rhamnode concentration. However, I do want to highlight that with our low and high rhamnode concentrations, there was no comparable difference in DNA content in comparison to the control, meaning that our treatments, our low and our high rhamnode, did not significantly decrease the living DNA content. And so now that we've looked at the biocompatibility of our treatment, we wanted to pose another question. Can we implement our rhamnolipids into a wound healing device to sequester uranium in the wound bed? And here I have the general process of electrospinning, which is a tissue engineering technique where we implement a protein polymer solution. It undergoes a high voltage and spits out these electrospun nanofibers, creating a scaffold, which I have imaged there on the right. And scaffolds are used in wound healing as they promote cell migration and proliferation, and they're made to mimic the extracellular matrix. And we went ahead and seeded cells onto these scaffolds, and we wanted to see just how well they're able to migrate and weave into these structures after they've been impregnated with our treatment. And so we hypothesized that our neonatal fibroblast cells will continue to exhibit normal cell behavior and be able to colonize the scaffolds after they've been impregnated with our treatment. And so we did this by using scanning electron microscopy, which to qualitatively analyze our scaffolds with and without rhamnolipids. And here we have a control scaffold, meaning it has no rhamnolipids whatsoever. And we see just why it's so favorable environment for these cells, as it is made to mimic the porosity of natural extracellular matrix. And so we went ahead and imaged a scaffold that's been impregnated with a 2.5% of our treatment, and we see that there was no structural difference between our impregnated scaffolds and our control. And then we went ahead and cellularized and grew cells onto these scaffolds to demonstrate our impregnated scaffolds' cytocompatibility. And here I have highlighted in green and also indicated by those red arrows just how well those cells were able to weave and migrate into the structure even after being in the presence of our treatment. And so in summary, we used the cell migration assay to compare closure rates across different treatment groups and saw that even at that 12-hour mark with being exposed to uranium, there was still 100% closure. And then as well with our Presto Blue and Cyquant Direct assay, we measured biocompatibility between our cells and our treatment and saw that not only was there an excitement in metabolic activity with our low and high concentrations of our treatment, but there was no comparable difference in living DNA content between our treatment and the control cells. And as we saw with the SEM, that the structure of our impregnated scaffolds did not inhibit normal cell activity. And future work, I would love to see in vivo mouse studies. And so with this work, I hope that we can develop a healing therapeutic to help those infected by internal uranium toxicity. Thank you. Yeah, I do.
There were some pretty stark differences with your rhamnolipid treatments. Is there any literature that supports that rhamnolipids acts as an extracellular signal? Like, is it like a endocrine or paracrine juxtacrine signal naturally? Because you said it's biologically based, right? Mm-hmm. Yeah. Sure.
I know in literature it's used as like a surface, like a membrane, and like it's a membrane active molecule, basically, and it's been used in the soil to chelate uranium. So it has a wide range of applications. Yeah. Does that answer your question? Okay. Yeah.
What is the level of uranium that would induce the toxicity effects that you received earlier?
I'm sorry, can you repeat?
What is the uranium concentration required to induce the toxicity effect that you were listing earlier in terms of renal failure, the wound, etc.?
That's a great question. I know that in our lab, we do a 0.1 micromolar, and we've seen negative effects that way. So there is, yeah.
Have you measured uranium here? Because I saw your map. It's pretty close.
In our lab, no, but in other labs on campus, they have, yeah. So. We do a weight-by-weight concentration in our polymer protein solution. We do an overnight, we inject it with the Ramno, and then it's in the syringe, and that undergoes the voltage, and it's there in the scaffold, yeah. Yeah.
How do you evaluate the degree to which uranium has been sequestered in a scratch assay by the Brenda?
Right now, in the graph, we do percent closure rates. So even under the presence of uranium, we saw that those cells had an increase in percent closure rate in comparison to the controls of just uranium. So it's preliminary stuff, but it's a good indicator to start there. Yeah. Okay.